Hello, and welcome to today's edition of Talking to a Biologist. Today we're with Dr. Bruce Ford from the Department of Biological Sciences. And Bruce, tell us where we are.
Well, today we're in the University of Manitoba Herbarium, which is a collection of dried pressed plant specimens. And that may not sound too glamorous to some of you, but it's actually a very valuable teaching and research resource.
All right. You say it doesn't sound that glamorous. What got you interested in all of these dried plants?
Well, I first got interested, believe it or not, in plants through bird watching. And I was an amateur bird watcher for a number of years and got very intrigued in birds. And then I met somebody who was a very accomplished field botanist and decided to he got me interested in other groups of organisms, and that happened to be plants. And I found plants a little bit more challenging than birds, and that sort of got me where I am today.
So where are you from originally? You're here now in Manitoba. What was your academic career that led you here to the University of Manitoba?
Well, I grew up in Southern Ontario in the Toronto area, and I did my PhD at the University of Toronto. And from there I went to the Missouri Botanical Garden, where I worked on a project called the Flora of North America. And then from there I came to the University of Manitoba, and I've been here since 1993.
Since 1993. All right, so we're here in the herbarium. How many pressed plant specimens are there in the University of Manitoba?
We have in this collection here approximately 75,000 specimens, and this is the largest collection of dried pressed plant specimens in the province.
It's the largest in the province. And how would it compare to other universities? Are there large ones at other institutions?
Yes, we're certainly not the largest in the country. The National Museum, for example, has over 1 million specimens, as does the Department of Agriculture. University of Montreal is probably one of the largest in the country, and I can't remember. They're probably about 750,000 specimens.
Wow. So how broad or how close are we to representing all of the vascular flora of Manitoba in this collection?
We're actually pretty close. We obviously have some things that perhaps haven't been seen in a long, long time. Perhaps they're extinct or extirpated from the province, but by and large, we have a very good representation of most of the 1,500 or so species of vascular plants that occur in the province.
Now, if a student, after finishing their first year of biological sciences here, was more interested in these pressed plants and looking at Manitoba's biodiversity from the plant side, what sort of courses do we offer here at the U of M?
We offer a couple of courses. Probably the one that uses the collections the most here is my course called Vascular Flora of Manitoba, which is a course that, amongst other things, introduces you or provides you with the basic skills in plant identification. And hopefully by the end of the course you have enough wherewithal to determine what a particular species is and how it differs from other species in the province.
Now, compared to other jurisdictions in Canada, how is Manitoba doing in terms of tracking its biodiversity?
We're probably not doing too badly. This collection I mentioned sounds like it may be dry and boring at first, but it actually provides us with a detailed coverage of the plants that occur here in the province. So we have within these cabinets here documentation of where all these species occur, and they've been collected at multiple places, so we can actually do detailed mapping of where species might occur in the province. So we use this resource to track species. Some of them may be rare and endangered, so that becomes very useful. We also have, the province also has what's called the Canadian Data Centre, which also helps track rare and endangered plants in the province as well too. So we feed into a number of different groups that are interested in tracking plants in the province.
Very interesting. Now, when we dry these plants, are we still able to get their DNA if we want to do some DNA analysis as well?
Absolutely, and that's been one of the exciting things that's happened in the last 10, 15 years or so, is that these plants, as I said, look dry and boring, but actually we can get so much out of them. We can figure out where they came from in terms of their location. We can look at their morphological features, but what's really exciting is we can break off a little tiny fragment and we can extract DNA from those plants, some of them upwards of 50 years old, and we can get intact DNA from that. And then from that information, we can look at how they might be related to other species, and so there's some really exciting new possibilities that wouldn't have been imagined just 15, 20 years ago.
So maybe it'll be a new show, CSI Plant. Can you show us inside one of the cabinets?
Absolutely. So in this cabinet here, you can see we have what looks like a collection of folders. And it's in these folders that we keep our specimens. So do you want me to pull out one of these for you? So the folder that I've pulled out here is a folder that contains specimens of a tree that some of you might be familiar with. This is trembling aspen, which is found quite widely over a large part of the province. But you can see that the specimen is a bit of the plant. You can see here it's a twig and some leaves. And this would be similar to what you did in kindergarten with drying plants in a foam book. We're a little bit more complicated here. We have something called a plant press where we dry these in, and then they're mounted on archival paper, glued on that paper, and then you can notice down here in the corner we have a label which indicates the name of the species and detailed information from where it was collected. And the exciting thing is now, with our most recent collections, a lot of them have GPS coordinates on them. So we can now map them using satellite data and provide detailed documentation of where they occur precisely on the landscape. And as you can see, we have multiple specimens of the same species. So by having this coverage over a large area, we can provide detailed mapping for many species.
All right. Well, thank you very much, Dr. Ford. And that concludes today's episode of Talking to a Biologist, and your lecture will start now. Thank you.